This will leave you speechless. Terrence Howard is about to show you how the hidden rhythms of frequency shape our world. This is real, powerful, and it's all around us. So sit tight and let's dive in.
Our DNA is made up of what? 5 elements, right? Hydrogen, carbon, nitrogen, phosphorus, and oxygen. You know how they're arranged? The phosphorus sits right here in the middle, and you've got 4 oxygen atoms surrounding it. Then there's another phosphorus, 2 more oxygen atoms. Now these are the legs of that double helix. These are the legs. Now the latter steps on it, they become the adenine, the guanine, the cytosine, and thymine, the ATCGs. It turns out hydrogen, they used to think that energy, the body transferred or understood the energy information through electricity. They're finding out it's through frequency. Well, guess what hydrogen sounds like? It's a key of E, and it's a color yellow. That's what proper physics gets you to. Oxygen is a chartreuse, and it's the key of F over F sharp. Now they used to, like I said, information used to be thought about as just electrical information passing on. But I've got a question for you. Have you ever been at a party with some of your friends you ain't seen in 10 years, but y'all was like 14, you hung out and there's a song that came on and you was like, ah, and everybody got up and was live at that moment. Now say 10 years later you got a friend with you, he's a good guy, but he wasn't there when me and my boys formed this. So when that song comes on, our DNA tightens. There's a particular song, a scale in the genomes for each and every one of us. Now, when that song is played, when your particular prime resonant frequency is played, guess what happens? Your DNA celebrates and tightens. Everything else gets pushed out. Harmonic wave resequencing. The carbon is also the key of E and yellow. The nitrogen is G. And all these play a song. So say that the F over F sharps of the oxygen, the green, is only hitting at an F. Why? Because there's another frequency coming in from a generator over there. That's changing and causing it to change. The F hits the carbon. So instead of hitting as an F sharp, it hits as an F, so when it hits the carbon at an E, it doesn't hit it quite right, so the domino don't file right. So the next domino hits wrong. And so now we have malformation happening within our DNA because the frequencies are being pushed off. But when we hear our tone, we tighten back up. So yeah, I'm in the process of building the harmonic wave resequencer with another doctor who has been able to successfully cure AIDS and cancer. And it's well underway. But I wanted you guys to understand how that works. It's so simple. You do not need to tear the human body apart when all you can do is use frequency to bring it back to its normal space.
Terrence Howard's known as an actor. He starred in Hustle and Flow, Empire, but he also has a patent on technology that he's created in his own mind, and it's called Teriology. It's a logic language, which he called Teriology, and he kept it a secret until he was able to patent it. But I'm going to hand this over to the you only artificial intelligence help of the day, Roger. Thank you, Roger. Take it away for RNA and DNA.
Today, we're going to be talking about the only 2 types of nucleic acids that exist, DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. As nucleic acids, both DNA and RNA are long organic macromolecules, which means they are very large molecules that contain the element carbon. Both DNA and RNA are composed of nucleic acid monomers called nucleotides. Nucleotide monomers always contain a phosphate group, a 5-carbon sugar, sometimes referred to as a pentose, and a nitrogenous base. But DNA nucleotides have the 5-carbon sugar deoxyribose, while RNA has a different 5-carbon sugar called ribose. The pentose sugar in DNA has one less oxygen than ribose in RNA, which leads to its name deoxyribose. Let's focus on the nitrogenous bases in DNA for a moment. Each DNA nucleotide contains only one of 4 possible nitrogenous bases: adenine, guanine, thymine, and cytosine. These 4 bases are usually abbreviated A, G, T, and C. When nucleotide bases connect through hydrogen bonds, it's important to know that the bases always pair up with the same partners. Adenine only pairs with thymine, and guanine only pairs with cytosine. This is called the base pair rule. Scientists classify nitrogenous bases according to the shape of their molecules. Adenine and guanine both have a double ring structure containing a 6-membered and a 5-membered nitrogen-containing ring fused together. These types of ring structures are chemically classified as purines, but thymine and cytosine both only have a single 6-membered nitrogen-containing ring shape, so they are chemically classified as pyrimidines. Here's a little hint to help you remember this. Pyrimidine has a Y in its name, and so do the bases classified as pyrimidines, thymine, and cytosine. Like DNA, an RNA nucleotide may have the nitrogenous bases adenine, guanine, or cytosine, but unlike DNA, which has the nitrogenous base thymine, RNA has the nitrogenous base uracil instead. This means that any nucleic acid that contains thymine must be a strand of DNA, and that any nucleic acid that contains uracil must be a piece of RNA. Like thymine, uracil has a single nitrogen-containing ring, so it would also be classified as a pyrimidine. So, what do the nitrogenous bases have to do with building a DNA molecule? Well, each strand of DNA begins to assemble when the phosphate group in one nucleotide bonds with the 5-carbon sugar in another nucleotide. The 2 strands that make up a DNA molecule are linked through hydrogen bonds between their nitrogenous bases. When the spiral DNA molecule is untwisted, these base pairs look like rungs on a ladder. The spiral DNA molecule is described as having a double helix shape. James Watson and Francis Crick first described DNA's double-stranded helical structure in 1953, after looking at X-ray images of a DNA molecule taken by Rosalind Franklin. Now, let's look at the structure of RNA and see how it compares to the structure of DNA. While DNA is a double-stranded molecule, RNA is always single-stranded. In eukaryotic cells, DNA is found only in the nucleus. But RNA can be found in the cytoplasm as well as in the nucleus. DNA controls heredity by containing the code or set of instructions for building the functional and structural proteins that make up your body. And RNA uses these varied instructions to build all the proteins a living organism needs. There are 3 types of RNA: messenger RNA, referred to as mRNA, ribosomal RNA, referred to as rRNA, and transfer RNA, referred to as tRNA. Messenger RNA starts off in a cell's nucleus. Here it transcribes or copies the instructions for assembling a protein from a portion of the cell's DNA. Next, the messenger RNA takes these instructions or protein recipe to a ribosome in the cytoplasm. The ribosome, which is made out of ribosomal RNA and proteins, is the site where this recipe is read and translated into a protein. Finally, transfer RNA in the cytoplasm brings specific amino acids to the ribosome so that the particular protein encoded by the recipe can be made. DNA and RNA are the only 2 nucleic acids that exist. Both DNA and RNA are built from nucleic acid monomers called nucleotides.
Which we've talked about nucleotides in other podcasts I've had in the past.
Nucleotides always contain a phosphate group, a 5-carbon sugar, and a nitrogenous base. Deoxyribose is the sugar in DNA, and ribose is the sugar in RNA. The nitrogenous bases in DNA are adenine, thymine, cytosine, and guanine. The nitrogenous bases in RNA are adenine, uracil, cytosine, and guanine. In DNA, adenine always pairs together with thymine, and cytosine always pairs together with guanine. Adenine and guanine are purines. Cytosine, thymine, and uracil are pyrimidines. DNA is composed of 2 strands linked through hydrogen bonds between their nitrogenous bases. The 2 DNA strands are twisted into a double helix shape, first identified by Watson and Crick.
The Secret of Life, DNA, is the book we will be reading today by James Watson, which he just mentioned. So thank you for tuning into the You Only podcast. Now back to Roger.
RNA is a single-stranded molecule. In cells, DNA is found only in the nucleus, while RNA is found in the cytoplasm as well as the nucleus. DNA controls heredity by containing the instructions for building the proteins that make up an organism and allow it to function. RNA uses these instructions to build proteins,
Which you intake chicken, fish, eggs, milk. Those are the proteins your body needs, lean proteins, to help build those amino acids up.
There are 3 types of RNA: messenger RNA, ribosomal RNA, and transfer RNA.
Could you explain to the audience members what the difference is between messenger RNA, ribosomal RNA, and transfer RNA?
Messenger RNA copies the instructions for building a protein and takes them to a ribosome. Ribosomal RNA is the site where the instructions are translated into a protein. Transfer RNA brings specific amino acids to the ribosome so the correct proteins can be made.
And I have said in other podcasts that amino acids are so important, so your body has all the correct tools it needs for your DNA and your RNA. Like Terrence Howard said, they tighten. So you want your DNA to be tight, so then you're always sharp and apparent and coherent at all times during your time here on this planet, which is short. And thank you again to Roger, our artificial intelligence reader of the day. We do appreciate you here at the You Only podcast, and hopefully my fan members are going to enjoy Roger. He will be bringing us some facts and some updates here and there about RNA and DNA throughout this season. And we got a lot more to learn here at the Only You podcast. Today we're going to be doing James Watson and DNA, so hold tight.
Swing it like Roger.
As was normal for a Saturday morning, I got to work at Cambridge University's laboratory earlier than Francis Crick on February 28th, 1953. Listen up, you Only fan members. I had good reason for being up early. I knew that we were close. Had I any idea just how close to figuring out the structure of a then little molecule called DNA? This was not any old molecule. DNA, as Crick and I appreciated, holds the very key to the nature of living things. It stores the hereditary information that is passed on from one generation to the next, and it orchestrates the incredibly complex world of the cells. Figuring out its 3D structure, the molecule's architecture would, we hope, provide a glimpse of what Crick referred to, only half jokingly, as the secret of life. We had already knew that DNA molecules consist of multi-copies of a single basic unit, the nucleotide, which comes in 4 forms, and which we learned about those forms earlier. I had spent the previous afternoon making cardboard cutouts of these various components, and now, undisturbed on a quiet Saturday morning, I could shuffle around the pieces of the 3D jigsaw puzzle. How did they even fit together? Soon, I realized that a simple pairing scheme worked exquisitely well. It fitted neatly with T and G and C. Was it? Did the molecule consist of two chains linked together by AT and GC pairs? It was so simple, so elegant, that it almost had to be right. But I had made mistakes in the past, and before I could get too excited, my pairing scheme would have to survive the scrutiny of, yep, you guessed it, Mr. Crick's critical eye. It was an anxious wait, audience members. But I need not have worried, because Crick realized straightway that my pairing idea implied a double helix structure with two molecular chains running in opposite directions. Everything known about DNA and its properties, the facts we had been wrestling with as we tried to solve the problem, made sense in light of those gentle complementary twists. Most important, the way the molecule was organized immediately suggested solutions to two of biology's mysteries, and that was how hereditary information is stored and how it is replicated. Despite this, Crick's brag in the Eagle, the pub where we habitually ate lunch, that we had indeed discovered the secret of life, struck me as somewhat immodest, especially in England, where understatement is a way of life. Crick, however, was right. Our discovery put an end to a debate as old as the human species. Does life have some magical, mystical essence? Or is it like any chemical reaction carried out in a science class, the product of normal physical and chemical processes? Is there something divine at the heart of a cell that brings it to life? The double helix answered that question with a definitive no. Charles Darwin's theory of evolution, which showed how all of life is interrelated, was a major advancement in our understanding of the world in materialistic psychochemical therapy terms. The breakthroughs of biologists Theodore Swan and Louis Pasteur during the second half of the 19th century were also an important step forward. Rotting meat did not spontaneously yield maggots, rather, familiar biological agents and processes were related, in this case, egg-laying flies. The idea of spontaneous generation had been discredited. Despite these advancements, audience members, various forms of vitalism, that is, the belief that psychochemical processes cannot explain life and its processes, I do believe they linger on. Many biologists reluctant to accept natural selection as the sole determinant of the fate of evolutionary lineages invoked a poor divine overseeing spiritual force to account for adaptation, but physics, accustomed to dealing with a simple pared down world, a few particles, a few forces, found the messy complexity of biology bewildering. Maybe, they suggested, the processes at the heart of the cell, the one governing the basic of life, go beyond the familiar laws of physics and that of chemistry. Did we ever think of that, audience members? That is why the double helix was so important. It brought the Enlightenment's revolutionary and materialistic thinking into the cell, the mitochondria, the cytoplasm. We learned about that earlier, folks. The intellectual journey that had begun with the wonderful displacing humans from the center of the universe. The continued And that continued with Darwin's insistence that humans are merely modified monkeys had finally focused in on the very essence of life, and there was nothing special about it. The double helix is an elegant structure, but its message is downright prosaic. Life is simply a matter of chemistry. Crick and I were quick to grasp the intellectual significance of our discovery, but there was no way we could have foreseen the explosive impact of the double helix on science and society. Contained in the molecule's graceful curves, with the key of molecular biology, a new science whose progress over subsequently 50 years has been astonishing. Not only has it yielded a stunning array of insights into fundamental biological processes, but it is known for having an ever more profound impact on medicine, on agriculture, and the law. DNA is no longer a matter of interest only to white-coated scientists in obscure university laboratories. It affects us all. And I think every one of us knows that here at the You Only podcast. By the mid-60s, we had worked out the basic mechanics of the cell, and we knew how, via the genetic code, the 4-letter alphabet of DNA sequences translated into 20-letter alphabet of the proteins. The next explosive spurt in the new science growth came in the 70s with the introduction of techniques for manipulating DNA and reading its sequence of base pairs. We were no longer condemned to watch nature from the sidelines, but could actually tinker with the DNA of living organisms. And we could actually read life's basic script. Extraordinary. New scientific visas opened up. We would at last come to grips with genetic diseases from cystic fibrosis to cancer. We would revolutionize criminal justice through genetic fingerprinting methods. We would profoundly revise ideas about human organisms and human origins, about who we are and where we came from by using DNA-based approaches to prehistory, and we could improve agriculturally important species with an effectiveness we had previously only dreamed of. But the climax of the first 50 years of the DNA revolution came on Monday, June 26, 2000, with the announcement by U.S. President Bill Clinton of completion of the rough draft sequence of the human genome. Today we are learning the language in which God created life. With this profound new knowledge, humankind is on the verge of gaining immense new power to heal. The Genome Project was a coming of age for molecular biology. It had become big science with big money and big results. Not only was it extraordinary, technologically advanced, the amount of information mined from the human complement of 23 pairs of chromosomes is staggering, but it was also a landmark in terms of our idea of what it is to be human. It is our DNA that distinguishes us from other species, and that makes us the creative species, dominant, destructive creatures that we are. And here it is entirely what that set of DNA, the human instruction book. DNA has come a long way from the Saturday morning in Cambridge. However, it is also clear that the science of molecular biology, what DNA can do for us, still has a long way to go. Cancer has still to be cured. Effective genome therapies for genetic disorders still have to be developed. Genetic engineering still has to be realized its phenomenal potential for improving our food. But all these things will come. The first 50 years of DNA revolution witnessed a great deal of remarkable scientific progress as well as the initial application of that progress of human problems. The future will see many more scientific advancements, but increasingly the focus will be on DNA's ever greater impact on the way we live our life. And thank you for listening to the Only You podcast. And we're learning about James Watson's book, and he is a Nobel Peace Prize winner. And this book, DNA, The Secret of Life, is phenomenal. And I hope you rush out, check it out, buy it, and learn. You Only Season 3 podcast rebranded. You guys have showed up again to another one of my wonderful episodes. I'm going to try to do more episodes. I'm going to try to put more out, more content. I'm trying to make it more exciting. I'm trying to share some things with you that I've listened to and found that may change your thinking, make you understand things are different, each and every little country out there. And I have a lot of fan members in Australia. I have a lot of fan members in India. I'm grateful for all of you. I'm grateful for those podcast listeners in Egypt who rose You Only podcast to number 15 out there with all the crises that are going on around that country. I do pray for them. I hope that everything has worked out and that every person over there is given the freedom to express their religious beliefs, and that life is only temporary, and that if we could love, it would cover a multitude of sins. And thank you again for tuning into the You Only podcast. This is your host, Lo Jackson, and we are on a wonderful month here, and that's December. We're on December here, the last month of 2023, and it's a month of forgiveness, love, because love covers, like I said, a multitude of sins and forgivenesses, and love covers a multitude of questions that we are faced with daily in our lives. And many of us don't understand that some of our problems are from the DNA that we're given when two people make us, and we come out with both of their DNAs, and sometimes those DNAs mix well and sometimes they don't. And we learned a lot about that in this podcast earlier. And Seasonal Depression Awareness Month is what our awareness is at the You Only podcast. And I like to have an awareness just for the fact that many people go through each month and think they're alone, and there's nobody out there that has seasonal depression, or they're thinking that they're alone, because that's what it makes you feel like when they set back the clocks and the days turn into night quicker. Things change for those who have seasonal depression. They become very sad and alone and upset. I know I go through the winter blues hard in February, and it's one of the worst feelings ever. So I know what you're going through. I can relate. And I wanted to introduce you guys to a guy named Robert Levitan, and his name's Dr. Robert Levitan. He's a senior scientist and research head for mood and anxiety disorders, and he is a wonderful doctor, and he knows a lot about seasonal depression. And I want to turn this over to him. And what can you tell us about seasonal depression, Doc?
Right around the time that we changed the clocks, which is now in November, the days, of course, are getting very, very short. And many people start to experience a form of depression that we call seasonal depression or seasonal affective disorder, which often starts with fatigue, low energy, but starts to involve more symptoms of depression, symptoms of sadness, lethargy, apathy. So about 2 to 5% of Canadians will have a severe clinical form of seasonal depression. Another 10 or 15%, though, have a milder form. And then another large group of Canadians, perhaps as many as 25 to 35%, would have the winter blues, which is just a sense that things aren't quite as positive in the wintertime as they are in the spring or summer. Seasonal depression is about 80% in women and only 20% in men. When one starts to treat seasonal depression, there are 3 options that are commonly used. The most common being light therapy, the second being antidepressant medication, and another option being tryptophan, which is a natural substance that is available in pill form. Now, many people will have milder forms of seasonal mood change without having true depression. The same strategies work in that case. Particularly light therapy is often used for people with milder forms of seasonal depression. Increasing exposure to natural light is very beneficial if it is feasible, but in reality it is often difficult to predict the quality of light in the wintertime, and it's often not realistic that people are going to consistently be able to get the natural light that's required to reverse the illness, and therefore the existence of light therapy units that are specifically designed for treatment of the disorder. I get asked a lot about exercise, and I would see exercise more as a preventative or maintenance treatment once people are better. So on a daily basis, we hear about the epidemic of obesity, and interestingly enough, seasonal depression is a contributor to this problem in a northern climate such as Canada. Ultimately, one needs to accept that there's likely to be some weight gain each fall-winter period. The key thing is to try to limit it as much as possible using healthy choices as well as treatment of depression where necessary. The key point today is to make sure that if you're feeling signs of depression this time of year, going into the winter period, make sure to first get assessed by a doctor before confirming the diagnosis and going on to treatment options.
Thank you again to Dr. Robert Levitan for that wonderful rundown of seasonal depression, because it is Seasonal Depression Awareness Month at the You Only podcast, because you only are the ones that can learn and be aware of your situation. If you have these things going on, go to your doctor, get checked out, and maybe you can get on a regimen and feel a lot better throughout the years instead of always feeling like doom and gloom and suffering. Thank you again for tuning into the You Only podcast, and hopefully you've enjoyed James Watson, RNA, DNA. I love teaching and learning more about that stuff, and there'll be more stuff to come that I'm going to build off of this episode, and hopefully you'll go back and check out Edward Ross, Social Control, that I had done yesterday and John Taylor Gatto's take on that. Enjoy the podcast and feel free to join in on the polls and the questions that I leave there at Spotify, because your feedback is what helps build a better podcast. So then I can kind of adjust the situation and include more things. And, I am still learning how to edit, mix, and make it more interesting and fun for the audience members who do come here every week and seriously support my podcast. I'm grateful. I don't know everybody that listens, but I know some people that listen, and I am grateful for all of you, and hopefully we can learn together and we can stay connected. And thanks again. I do appreciate every single one of you, and I do mean that.